Well, today I was on a breakfast panel talking about imposter syndrome, and right now, this afternoon, I'm talking to food scientists, and I'm a mere virologist, so I don't know anything about food safety. But hopefully what I'll talk about is something that could be applied to food safety and maybe should be applied. So usually, I mean, if you've seen me speaking over the last few years, it's probably to do with COVID genomics, but I'll spare you that today, but don't worry, because the next pandemic is just a few mutations away. There's currently a pandemic among wild aquatic birds, and it's sweeping the world. It's been circulating for a few years now. It's a highly pathogenic strain of an avian influenza virus. And this virus has killed thousands of wild birds, and it's had multiple spillovers to mammalian hosts as well. In New Zealand, if or when this virus comes here, it would have a devastating effect not only on industries but on wildlife, particularly on wild aquatic native taonga species such as hoiho or yellow-eyed penguin. The yellow-eyed penguin is something that I've been working on in collaboration with a whole bunch of people recently because they have many diseases that is threatening their extinction from the mainland of New Zealand. In particular, I'm going to talk about two different diseases that have been affecting this species and the use of genomics that has helped us identify possible causative pathogens. So here's a distraction of really cute chicks from the Wildlife Hospital in Dunedin that I work with collaboratively, just so that nobody asks me about causation, because molecular genetics can only go so far. So one of the diseases affects really young chicks. They usually die within a week of life, and in fact, over the last few years, this has affected about a quarter of chicks that hatch die of this diphtheric avian disease. This has been noted just over the last couple of years, and we think that it might be 100% fatal, although we only see it when they die. So it is a hemorrhagic disease affecting lungs. Basically, the lungs fill with blood, and they die within hours of presenting clinically with this disease. So we worked with the Yellow-Eyed Penguin Trust as well as the Wildlife Hospital that monitor these chicks, and they are monitored at 3 different population sites around Otago. And we did next generation sequencing or metatranscriptomics, which I'll explain in a little bit, and found just one virus that was present really abundantly in these tissues of these dead chicks. And this was a novel gyrovirus. Now, if you're in the poultry industry, you might know of a gyrovirus called chicken anemia virus, and which affects chicken, poultry, and chicks in the same sort of way, in a different sort of disease, but it can have similarities. And so we found not only very abundant whole genomes of this virus in chicks, but we found a whole sort of diversity of this virus, suggesting that it's been circulating for quite a long time, perhaps asymptomatically, and in much more prevalent than we think that it is presented clinically. Another disease affecting hoiho chicks is a disease that has been circulating in these chicks for about 20 years now. And this is a diphtheric disease, and it basically presents as oral lesions in the oral cavity, stopping them from feeding, and usually they starve to death. And there has been a lot of interest in this disease because of the high morbidity and mortality it causes. And there has been, in the past, some sort of culture-based work done to detect the presence of different bacteria species. However, we did some, again, metatranscriptomics and found there was no sort of relationship between the bacteria, other microorganisms, between different stages of the disease or healthy adults. But what we did find is a really highly abundant novel virus called virus. This is a virus that sits in a genus called megavirus in the Picornaviridae family, so it's a picornavirus. These things include things like hand, foot, and mouth disease that you might be familiar with as well. So here's another distraction for you because, again, do not ask me about causation, but what we did find is other viruses in this genus affect avian species, cause disease, especially things that cause disease affecting beaks as well. And so this presents, a potential pathogen that could be studied further to understand the causation. And so for these investigations, what we do is take a whole infectome approach, where we're trying not to bias ourselves into one type of organism. So if we, use virus culture or PCR, and these have their places, but they're not very good at identifying novel things because you kind of need to know what you're looking for. But when we take a metagenomics or a metatranscriptomics approach, we can then look at this metaphorical tree of all microorganisms or all potential pathogens and identify the whole picture. The problem is that our sampling has been extremely biased. Up until now, we've really only been interested in viruses or pathogens that affect us, or plants and animals that are important to us, usually economically important. So our knowledge of what is a pathogen is very biased. And so what we do in my research lab is try and understand the diversity of the virus sphere across the tree of life, because it's not only by understanding what might be out there in nature, we can then understand what might be causing disease. For example, Aotearoa has a very unique set of flora and fauna. And while bats are one of the most studied organisms for their viruses, they have been quite understudied in New Zealand, although there has been some preliminary work done by Richard Hall at ESR some time ago, who really spearheaded sort of virus hunting. So we, in collaboration with the Department of Conservation and others, we were able to sample both New Zealand bat species. And these have very different, unique, diverse life histories and evolutionary histories as well, where the lesser short-tailed bat has probably been evolving in isolation for about 35 million years, if not longer, where the long-tailed bat migrated from Australia much more recently. And by looking at the total infectome and concentrating here on just the viruses, we found that each have lots of viruses, but the only one they shared is a few of viruses within the Coronaviridae. These are coronaviruses. You've probably heard of these coronaviruses recently. So these are alpha coronaviruses, different from the beta coronaviruses that causes COVID-19. But interestingly, both bat species have the same virus, meaning that this virus has jumped from these hosts within the past million years or so. What's also interesting is that one of these viruses we found in these bats may represent the shortest known coronavirus genome to exist that is known yet. For example, we found a virus that has this whole deletion in its promoter region. For example, here is a deletion of several genes. We don't know what these genes really do yet, and we don't know why it might even function without these genes, but it clearly does. So by sampling these unique hosts, we can try and understand and broaden our knowledge of these diverse viruses. So, and the more viruses we discover, the more host jumping we detect. For example, before 2020, this was kind of the diversity of the coronaviruses that we knew about. We knew that they infected mainly mammals and avian species. However, through a collaboration, I was involved in lamprey reddening syndrome investigation, or kanakana. Kanakana are a very important kai for Maori, very culturally important, and they unfortunately suffer this disease, again a hemorrhagic disease called kanakana reddening syndrome. And when I was involved in trying to understand what might cause this disease, and we found a novel coronavirus in these lamprey. Again, don't know if it's actually causing the disease, but what this does tell us is, again, this was most likely a recent host jump from this virus into lamprey from other fish species. And by identifying this virus in Kanakana, we were also able to identify similar genetic relatives of this virus in a whole bunch of different fish and amphibians as well. And which really tells us the sort of breadth of coronavirus diversity. They don't all cause disease, and they're in seemingly healthy fish amphibians as well. And if you know anything about vertebrate evolution, you know that Kanakana are a basal outgroup for vertebrates, and so this in-group in this phylogenetic tree suggests a recent host jump. So at the broad scale, we see, every virus family that we have, we thought that only infected mammals and birds. The more we sample outside of those host classes, the more we detect in other host classes, or these sort of lower vertebrates. For example, when we went to a fish market in Sydney, we sampled seemingly healthy fish and found a plethora of viruses in every virus family that we thought only infected mammals and birds, including filoviridae, so these are Ebola viruses in fish, and influenza viruses in fish, and coronaviruses in fish. These are seemingly healthy fish. There is no risk or disease of them to transfer these viruses to humans. They are very different cells that they infect, and they seemingly just live commensally with these fish. So I guess what the take-home message from this work is really to try and understand the breadth of the virus sphere, and fish are a really interesting way to study the evolution of viruses. And so one of my PhD students was able to fish in a really remote location on the Chatham Islands, across 16 lakes and water bodies across the islands, to try and understand these really remote ecosystems and that are free from sort of introduced species, to try and understand what kind of viruses that they might have. And we were able to sample a range of different fish species from across these lakes. And interestingly, we found lots and lots of different viruses, which is not surprising. We were trying to understand what might drive this diversity of viruses, whether it be sort of seawater influence or community diversity of different fish species, or the food web, for example. But what we found was smelt that we found pretty much everywhere on these islands. Their viruses were remarkably homogeneous across the lakes, and so these viruses have likely been evolving with these species for millions of years. And again, just to reiterate that how little we know about disease-causing viruses is that we found in tuna, in long and short fin eels, this really divergent virus called Tossoviridae. There's only been one virus in this whole family that has been identified prior to us finding it in tuna, and that was in really severely diseased turtles that were found in Florida. So this disease-causing virus that was found in Florida about 10 years ago had no other genetic relatives in this virus family, really, really novel virus. And we found it in the Chatham Islands in long and short fin eels, not causing disease. So this really just reiterates how little we know about the virus sphere and the diversity of viruses that exist. And if we want to understand what does cause disease, we need to understand what's out there in the first place. So our virological data is misleading, and you probably can't read this, but this just shows how little we know about what causes disease. So in 2008, this was the sort of proportion of viruses that we knew caused disease. After lots of virological sampling through metatranscriptomics, this is 2020, for example, this is the proportion of viruses that we think cause diseases. So the more we sample the virus sphere, the fewer viruses we think cause disease and are just out there. And what they're doing, we don't know. So we need a lot more effort to try and understand the depth and breadth of these viruses. Thank you.
Any questions? Anna?
Wait for the microphone or...
This one behind you, Glenda, can get...
So this seems to reflect a lot what you see in the bacteriophage world, where there's loads of things that we never looked for, so we never knew about them. When you look at your virus sequences that you get out of that, can you actually find genes that, for instance, do host specificity? So if you actually analyze the sequences rather than the linkage with the host, can you actually find out, can you predict almost like what, if you find the virus, where it should be in?
That's a good question. I mean, viruses obviously use their host cellular machinery for entry and replication. The problem is that often there's so few studies in these viruses. There's often very divergent, highly genetically divergent viruses that there's genes there, but we don't know what they do. So we need to kind of, I guess, explore the diversity of the viruses to find these diseases, to find these genes and to find similarities between the genes and what they do in certain hosts. And there's a lot known about certain viruses in model species, but doesn't tend to extend to things like wildlife and other things.
Kia ora.
Kia ora.
I'm not a scientist, but I was just wondering about dis-ease, the word dis-ease and imbalance of environment, and the environment's need to rebalance itself, or the imbalance creates these diseases. So just wondering, how far back are you able to trace the evolution of the virus? Is it, as far as being an Aotearoa, can you trace it back to, say, pre-colonization?
Yes, for sure. So in the virus world, we're looking at hosts that have been around for 500 million years, for example, in the case of Kanakana, likely evolved around that time. So that's the sort of time scale we're looking at. So we can look over millions of years, and we can look over weeks. What you're saying about the environment is absolutely really important. We don't really know what causes disease and why a pathogen or a microorganism is a pathogen in some hosts and seemingly commensal in other hosts. And in some hosts where it does cause disease, it may be an environmental trigger that made that disease take place. So.
Thank you.
No worries.
Kia ora, Jemma. Clearly not an imposter. You did very well this morning at breakfast. I'm not sure if I misunderstood this, but on one of your slides, it looked like there were 3 species at the top that had no viruses associated with them, mudfish and a couple of others. Is that unexpected, or?
Well, I guess our sampling size was necessarily relatively small, and so because we couldn't detect viruses doesn't mean that they're there. And what I didn't talk about is the sort of limitations of this unbiased way of sampling microorganisms, because what we're doing is, I guess, we've got a reference database, and that is all the genetic sequences that are known, and they are on reference databases, and that's what we can use to compare to our amino acids or whatever we've got, right? But the huge limitation in this work is that if something doesn't share sequence similarity with our genetic material that we've generated, we cannot recognize it, right? And there is this whole gray matter of probably lots of viruses that we just don't have the computational technology to identify.
That makes sense. Thank you.
That's it? Okay. Thank you so much, Gemma, and please join me by thanking you to all of our speakers today.